Hi folks, today we're going to be talking about enzymes in chapter 20. So enzymes, up until this point, we've had three different types of macromolecules that we've discussed. We've talked about carbohydrates, lipids, and proteins. Enzymes are proteins. They're just special kinds of proteins. We know that proteins have structural components to them, but we also know that some catalyze reactions. So when a protein catalyzes a reaction, particularly when we're talking in a physiological sense, these are called enzymes. And when we talk about catalyzing a reaction, what we're talking about is how easy or energetically facile it is for a reaction to occur. We know that if we have reactants, and if we took the sum of all the energy of those reactants and we had products, that if the reactants are higher in energy and the products are lower in energy, that the reaction will occur. That nature is inherently lazy, so it's always going to try and get to the lowest energy state possible. However, sometimes along the way there's a hump, and that hump is what we call activation energy. So while the product might be lower in energy than the reactants, there may be a huge energetic kind of like tax that has to go in to the process to get us going. Catalysis or catalysts help lower that energy. They lower the activation energy hump, making it an easier energetic process to go from reactant to product. So that is what an enzyme does. Whoops, sorry. So different enzymes act on different things. Enzymes have specific reactions that they do. So we can have reactions that will only occur on one substance, an enzyme that only works this one substance. Whenever an enzyme works on a molecule, another protein, anything like that, we call that a substrate. So there are enzymes that are like the little machine that does the work, and then there's the substrate, the thing that's getting work done to it. So we have different types of specificity to this. Absolute specificity would mean that the enzyme only works on one substrate, only on one substance. Relative specificity would mean that it reacts on a number of substances that are all related to each other. So they might physically have the same shape or all be in the same class or something like that. And then we can even have stereochemical specificity. So we could have an enzyme that distinguishes between the D and the L forms of something, or the alpha and beta forms of something. So here's an example. Here's urea with some water around. The enzyme urease is the enzyme that acts on the substrate urea to break down urea into two amino groups and some carbon dioxide. Now, enzymes, as we've mentioned, are kind of like these little machines that can be turned on or off. And turning an enzyme on or off is a term for calling it regulation. So we can regulate how often is the enzyme working, how often is it not working, what triggers it to turn on or what triggers it to turn off. And there are a number of different ways that enzymes are regulated. Sometimes the cell controls the rate of the reaction, or even sometimes we have controls where the product that is formed from the enzyme is actually also the trigger that turns the enzyme off. So it just depends on what we're talking about. Now, just like we've learned in all of Unit 2, we have this concept of naming, nomenclature, that there's a very organized way to name enzymes or anything. And so enzymes are no different. Just like we had common names and then we had IUPAC names for Unit 2, older names of enzymes and tend to add in IN to indicate that there's like a protein kind of aspect to them. So you see this in things like enzymes like pepsin, trypsin, chromatrypsin, things like this. However, we do have a more regulated naming system for enzymes now. So many enzymes now kind of follow this enzymatic commission system, nomenclature system. We have 6 basic classes of enzymes and the types of reactions that are catalyzed by those enzymes. And then within that, we talk about what type of substrate is used and what kind of reaction that it catalyzes. And then lastly, enzymes will end in the name ACE. So the root of the enzyme name ends in ACE. We already talked about this, but right, a substrate is the thing that goes under change, right, that is going to be worked on by the enzyme. So for example here's our urea again, and here is the water, right? So we called this urease before. The full name for it would be urea amido hydrolase. And what this means is urea is the substrate, amido is the functional group that is being formed, and it's being hydrolyzed. So we're cleaving this bond, and we're cleaving this bond, and the amide bond, amido, is being cleaved, hydrolyzed. So urea amido hydrolase. In this case, the substrate group was the urea, the functional group is the amide, and the type of reaction would be hydrolysis. Obviously, these names can get kind of big, so sometimes we just shorten them. What typically ends up happening is we will just add ACE to the ending of the substrate. Sometimes we also will just add ACE to a little combination of the substrate and the reaction. So there's a little more fast and loose going on with the naming here, but be comfortable with knowing if you see a name that you would be able to tell me something about the enzyme, either what type of reaction it does or what type of substrate it acts upon. So for example, we already saw urease as a shortened name. We also have something called alcohol dehydrogenase, which would be the functional group of alcohol or a hydroxyl, and we're dehydrogenating it. So here are those 6 classes that we talked about originally. Pretty much all the types of work that an enzyme can do would fall under one of these categories, and obviously there are tons of subcategories under this, but these 6 classes kind of encompass all the types of different work that an enzyme can do. So one of the ways that an enzyme can be regulated is with the use of cofactors. So sometimes an enzyme will require like a special second substance in order to be turned on. It's only going to be kind of weakly associated with it, so it can be popped on and off. If the second substance is another protein, we call it a coenzyme. So if it's an organic molecule of some sort, we would call that a coenzyme. If it's a non-organic molecule, like an ion or something like that, or non-protein, then we would call that a cofactor. Many of the enzymes in our bodies require cofactors. So a lot of them also require metal. So metal ions are a very important cofactor for many of the enzymes in our body. About 30% of the enzymes in our body actually utilize a metal cofactor. When the cofactor or coenzyme is not present, the enzyme is called an apoenzyme, and basically that means it's not turned on yet. It won't be turned on until the cofactor is added. When the cofactor is removed, it becomes the apoenzyme again. So we have apoenzyme plus cofactor, or coenzyme, whatever we're going to call it, turns it into an active enzyme. Vitamins are another very good example of these coenzymes. So this is why we need vitamins in our body. They're essentially the keys that turn on the engine to the enzyme. Here's a list of some vitamins and the form that they take and the function for their coenzyme. So a lot of these, again, it might not seem significant, but hydrogen transfer is a huge thing within our bodies. This can be anything from breaking apart double bonds into single bonds, moving around of hydrogen in terms of changing something from D to L, anything like that. We also have it as part of digestion, respiration. Hydrogen transfer is a huge one. So, how do enzymes work? We're going to talk about this in a very broad sense in the beginning. And really, what we're going to talk about is the active site of an enzyme. So, an enzyme we can think of as kind of like this blob that might have a little pocket or something in it, and that's where the substrate would come sit, and that's where the business end of the enzyme is. That's where the chemistry is going to occur. We call this the active site, where the enzyme and substrate bind together. When we have the enzyme and substrate bound together, we call that a complex. So we can have enzyme that is empty and a substrate that is floating around. When the substrate comes and binds to the enzyme, we get an enzyme substrate complex. Catalysis and chemistry occur. The enzyme then releases what is now a product, and the enzyme is ready to go and turn over another compound. So the substrate goes in and product comes out, but the enzyme goes in and the enzyme comes out. Just like any other catalyst, a catalyst is not changed in a chemical reaction. It comes out the same way it went in. For example, we have sucrase, which is an enzyme that works at digesting or breaking apart the disaccharide sucrose. Sucrose would be our substrate. So when the sucrose binds to the sucrase, chemistry will occur to cleave the glycosidic linkage between the glucose and the fructose. When that happens, the sucrase, the enzyme, will release the new products of glucose and fructose. There's no more sucrose left. The sucrose was broken down into the glucose and the fructose. There are two main fields of thought on how active sites work. One idea is that of a lock and key. So in this case, the enzyme would be the lock and the substrate would be the key. And that there's a perfect fit here, that the type of substrate will only fit in certain enzymes. And this is kind of a way to help explain concepts of specificity, that we have a rigid enzyme and that the shape of the substrate has to match the hole in the active site to be able to fit, to ensure that only certain substrates will be worked upon. The other concept is that of an induced fit theory, and this would be an idea that the enzyme has a general pocket, but it's soft and flexible, and that it will fit to match the shape of the substrate that it's working upon. So enzyme activity is a term we use to describe the rate at which the enzyme is catalyzing reactions. You've already heard me use the term turnover, but this is a concept of the number. When we say turnover number, what we're saying is the number of substrate molecules that one enzyme can act on in a minute. So obviously within our body, there's not just one sucrase enzyme or one urease enzyme. There's a concentration of them, but their turnover number would be the same depending on the conditions that are going on. Lastly, you may hear the term assay, an enzymatic assay, and this is simply an experiment that is run in a lab to measure the activity of an enzyme. So turnover numbers for enzymes vary greatly. Some go very, very slow, some go very, very fast. Remember, this is number of substrates that get acted upon or turned over by a single enzyme in 1 minute. So carbanic anhydrase, in 1 minute, one enzyme of carbanic anhydrase will catalyze this reaction 36 million times. However, DNA polymerase, adding a nucleotide to the monomer of the DNA chain, right? Only 900 of those will occur in a minute versus 36 million. So obviously this is a little more complex and goes much slower. This is a pretty rapid reaction that is being catalyzed here. So depending on the enzyme, the turnover number may vary greatly, but that doesn't necessarily mean that that enzyme isn't working properly. There's a wide variety of enzymes and rates at which they occur. So there are factors that can affect the activity of an enzyme, or its ability to perform at its best ability that it could. Obviously, the first thing would be the concentration of the enzyme. So if we have more enzyme around, we are able to turn over more substrate in 1 minute. So it would make sense that we have a direct relationship here, that as the increase the concentration of the enzyme, the rate of turnover for that enzyme would increase as well. We can increase substrate concentration, which would increase the reaction rate until it becomes saturated. So, this is actually the wrong image. This one should go flat. So instead of it dropping off here, think of this as coming out flat. So as we increase the substrate, so let's say I have 3 enzymes that will turn over a substrate. If I only have 1 substrate present, I have 2 enzymes that aren't working. As I increase the number of substrate concentration, now I'm going to be able to turn over more. So as I go from 1 substrate to 2 substrates to 3 substrates, I increased. But at 3 substrates, it will level out. So again, sorry, this is the wrong image. This will level out. And what that means is I can only turn over 3 substrates at a time because I only have so many enzymes. So the amount of enzyme will eventually be the deciding factor in how quick the rate can go. The last way we can affect an enzyme that we're going to talk about would be, and its activity, would be temperature. Now this is the right slide for this. So temperature, obviously in a physiological sense, would probably be somewhere around our physiological temperature. Typically at very, very cold temperatures, enzymes move very slowly, as do most things. But as we start to warm up, the rate will increase because the activity of the enzyme will increase. However, there will come an optimum point, right? That peak temperature where it's supposed to work. Anything above that will start to decrease the rate again as we start to unravel our proteins. So as you might imagine, when you have a fever and you are sick, you are raising your body temperature, and this can be dangerous not only for the invading either virus or bacteria, but also to our own bodies as well. Obviously, very, very high temperatures can lead to things like deafness, loss of vision, seizures, all sorts of not very good things. And that is because once we've reached the peak of optimal temperature, proteins begin to actually break apart. We call this denaturation. So obviously we're not going to have very good activity of our enzyme if it's falling apart. Same thing can happen with pH. So most enzymes have a pH that is optimal for them, depending on where they are in the body. If we go too low or too high in either direction, again, we can decrease the rate because we will begin to unravel the protein. So as I mentioned, there's obviously a very wide range of optimum pHs. It just kind of depends on the function of the enzyme and where it is supposed to be. So pepsin is a part of digestion in our bodies. So the optimum pH for that is around 1.5. However, when we get to something like arginase, which is found in like liver for carbonic anhydrase and things like that, the optimum pH is 9. So we have a huge swing in pHs here, and it's just dependent on the enzyme and the function of that enzyme. Another aspect of enzymes that can affect its activity would be inhibitors. Some inhibitors are natural, some inhibitors are things that are made particularly by us. A lot of poisons we consider inhibitors. A lot of antibiotics or other drugs are also considered inhibitors. You may be thinking, why would I want to inhibit an enzyme? But you have to remember that not only humans and animals have enzymes in them. So bacteria have enzymes in them. Viruses have enzymes in them. So if we can target bacterial enzymes, we have powerful antibiotics on our hand. So there are different kinds of inhibitors. The first kind we're going to talk about are what we call irreversible inhibitors, also sometimes called suicide inhibitors. These inhibitors will permanently bind to the enzyme at the active site most of the time, rendering it inactive. It will no longer be able to accept substrate and has essentially been turned off. So things like cyanide, mercury, lead, these are examples of irreversible inhibitors. So when someone experiences lead poisoning, there's no cure for lead poisoning. The damage that is done and can never be reversed. However, what we can do is try and stop further damage from occurring. Again, many antibiotics are irreversible inhibitors as well. Sulfa-containing drugs, penicillins, other related drugs all inhibit specific enzymes that are essential to the life processes of bacteria. This means that the enzyme that the antibiotic goes after stops the bacteria from being able to produce, hence stopping the infection. So for example, here are the general structures of penicillin. So over time, penicillin has been constantly kind of played with to keep up with resistance that we get. But here you can see many of the different forms of penicillin come from just changing this group here into one of these. Otherwise, the rest of the penicillin molecule remains the same. And so this would be a class of inhibitors that all are similar to each other, but we know over time that we get resistance to particular penicillins, and we need to come up with slight variations that the mutations in the enzymes have not overcome yet. So we said cyanide was one of these irreversible inhibitors. Obviously, cyanide is a very toxic poison. It acts very rapidly. Side note, if you've ever ingested enzyme, it tastes like almonds, very bitter almonds. What it does is interfere with the iron cytochrome oxidase, forming a very stable complex. So the iron in our body and cytochromes it's an iron 3 ion. And this needs to be able to, this iron is used to kind of complex with oxygen as it flows through our blood during cellular respiration. However, what will happen is the cyanide will bind very, very tightly to the iron. And when it does that, this iron is no longer available to bind with oxygen during cellular respiration. So you can imagine that you will essentially suffocate due to cyanide poisoning. The antidote obviously occurs within minutes if there's enough of it, but it is possible if exposed to it that we can have something called sodium thiosulfate. And what it does is turn cyanide into thiocyanate, which then does not bind as tightly to the iron and allows it not to poison the cytochrome. However, it's not something that can, once this has happened, even with the antidote, quote unquote, any of them that have been bound to the iron will no longer be functional. This won't turn into thiocyanate afterwards. So once some damage is done, that is it. There's no going back. But delivering the administering the antidote quickly will allow the antidote to scavenge for any free cyanide still floating around before they have a chance to complex with the cytochrome oxidase. Another very dangerous type of irreversible inhibitor are heavy metals. Heavy metals we know can poison you as well, so things like mercury, lead, right, things like that. And what ends up happening is that these heavy metals will do a similar thing that the cyanide does. So in our bodies, we have proteins, right, that have disulfide bridges on them or thiols and things like that. And oftentimes what will end up happening is something like mercury or lead will very tightly bind to these thiols instead of allowing disulfide bridging to occur. So what can end up happening is we have an active enzyme, and when either lead or mercury come around, they will bind very, very tightly and chelate to the lead or the mercury, and basically turn off the activity as that enzyme. Now, a lot of thiol-containing enzymes are part of neurological chemistry, so obviously this is one of those things that we associate lead poisoning with developmental issues, particularly in children. Again, just like we saw with the cyanide, there is no way to um, reverse any damage that is currently done. However, there are treatments where we have basically metal scavengers that will go through and hold tightly to the free metal in your body before it can get chelated to anything else, to any sulfites or anything like in our body. One of the very popular ones to do this with is EDTA. You will see EDTA in a number of different food products, everything like that. It is quite safe, but it will chelate a heavy metal. When delivered intravenously, it will chelate to the metal, and then like calcium ions, which are displaced by the heavy metal ions, are bound and chelate even more tightly. And so that complex EDTA with the heavy metal can then be excreted through our urine. So there are also reversible inhibitors. So irreversible inhibitors or suicide inhibitors, we often associate with something bad or something good if it's a therapeutic, and we're talking about something like an irreversible inhibitor on a bacterium. Reversible inhibitors are actually one of the ways that our body helps regulate enzymes. So there are reversible inhibitors, and those can be competitive or non-competitive. Reversible inhibitors obviously can bind and unbind from the enzyme, but there are two different kinds. There are competitive inhibitors, which compete with the substrate for the active site, and non-competitive inhibitors, which will bind somewhere else to the active site and bind somewhere else on the enzyme, changing the active site. So many drugs follow this competitive inhibitor path, and the idea here is that an inhibitor is designed to look as similar to the substrate as possible while not allowing the substrate to enter the active site and therefore be turned over. Zymogens, or also called proenzymes, are kind of a inactive precursor to an enzyme, and so as the enzyme is being produced, sometimes there's like a cap on the enzyme that needs to be cleaved off before the enzyme can be functional. And again, this is another form of enzyme regulation that can occur. So here we can see a bunch of different examples of zymogens and what their active enzyme is and what that process would be. So again, this is a very helpful way that physiologically enzymes can be regulated to make sure that they're not just constantly working and maybe doing more damage than they should, so that they can be turned off and be ready to be turned on at a determined signal. Allosteric regulation, or allosteric enzyme regulation, activity regulation, is the alternative bonding of a modulator. So a modulator is going to be something that will increase allosteric enzyme activity, right? So we're going to increase the activity of the enzyme. If we decrease it, that would be an inhibitor. So a modulator will increase activity, a inhibitor will decrease activity. We talked about this earlier as one of the ways to regulate an enzyme, but feedback inhibition is a way in which is an example of a modulator decreasing the activity of an allosteric enzyme. So what that means is that as the product is formed, that product is the trigger to turn off the enzyme or decrease the activity of the enzyme. We can see this in an example of threonine deaminase. So this is just the first step of it, but we have here, right, here's threonine, the amino acid, and it's going to go through this process when it goes through a downstream process of multiple enzymes But what ends up happening is the final product is isoleucine. Isoleucine happens to just be the cofactor that will inhibit threonine deaminase. So what it allows it to do is kind of stop the processing from occurring any further once the desired product has been made. Enzyme induction is the synthesis of an enzyme in response to a temporary need from a cell. So sometimes we call this a genetic control. So there will be a trigger by the cell to say, hey, we have a lot of this going on right now. Like we've just gotten a lot of lactose. So we need to break it apart. So we're going to make some beta D galactose that will help break up our galactose into galactose and glucose. That clearly, that type of enzyme doesn't need to be running all the time, only just in the presence of dairy. So we have triggers to help trigger those things and then turn them off once they've been used. Here's a nice flow chart kind of going through the basic steps of the different types of inhibitors that we see. Enzymes, oftentimes, misregulation of enzymes account for a very large number of the maladies that we experience as humans. Anything from being turned on when they're supposed to be turned off, being turned off when they're supposed to be turning on, overturning too many substrates, things like that. These all are uncontrolled regulations and can lead to other issues like uncontrolled growth of cells, which would be cancerous. Development of enzyme concentration in the blood serum is a major diagnostic tool. So if enzymes aren't supposed to leave, just like proteins aren't supposed to leave, so their specific cell, unless they are designed to travel. So if we find enzymes in a blood screening of the heart where they shouldn't be, or the liver or pancreas, these are good signs that we have damage to these organs, and that further studies need to be done. So these are some of the enzymes that we look for in blood panels when we are concerned about certain different ailments. So prostate cancer, bone or liver disease, heart attack, hepatitis, acute pancreatitis. These are all basic blood panels that can be run to figure out if we are having trouble with any of these areas. We have medical applications to enzymes as well. Isoenzymes are slightly different forms of the same enzyme that produce different tissues. So, serum levels of isoenzymes can be used for a number of diagnostic tools. But basically, if we have just slight differences in them, we're able to use them to distinguish between either a heart muscle and another type of muscle. And what this allows us to do is do distributions. So we know what we should have, and if we don't have these, then we know that we have a problem and need to do further investigations. All right, this is the end of chapter 20. Please make sure you're working on your homework. Let me know if you have any questions and are going over your reading.